Hello, everyone. Today we're going to talk about microarray analysis of gut microbiome gene expression in the presence of breast milk or formula in infants. So, a quick overview of what we're going to talk about today. We're going to talk about our gut microbiome, its omics history, the human gut microbiome and how it relates to our health, and then the development of this gut microbiome. Next, we're going to talk about breast milk versus formula, in which we show the different ingredients for both and how society today is debating the feeding between breastfeeding and formula. We'll also talk about our hypothesis and the questions we're going to ask ourselves today as we look over an analysis. And then we're also going to talk about microarray analysis, which is what is it and the experiment design that you do to perform this analysis. We're going to look into the NCBI gene expression Omnibus. We're going to go through an analysis. I'm going to show you how to look at everything step by step. And then we're going to analyze the results that we get by looking at this analysis. And then after that, we're going to ask ourselves what's next and how this analysis can influence for future research within the omics community and how the understanding of our gut microbiome expression in terms of disease development can occur. And then we'll just go over a quick little summary after all that. So let's talk about the omics history of gut microbiome. Up until the 1990s, gut microbiome testing was only limited to microbiological cultures. Once 1990 hit, we were able to see more next-gen sequencing to allow us to dive deeper into gut microbiome development. And then many techniques are based on a small subunit, ribosomal RNA or 16S rRNA, to do these experiments. Metagenomics, metabolomics, and phenomics also allow us to see the diversity of gut microbiome because there is so much gut microbiome within our guts that it allows us to see all the different types that are in there. And then in order to analyze gut microbiome, there's a lot of new techniques that have come up since the 1990s, and some include polymerase chain reaction or PCR, denaturing gradient gel electrophoresis, terminate restriction, fragment length polymorphism, fluorescence in situ hybridization. Then you analyze the 16S RNA-G sequencing. And there's two different sequencings on that. You can do one based on cloning, or you can do one based on amplicons. And then, of course, shotgun sequencing. So our gut microbiome, let's talk about it for a minute. So we know that it's composed of bacteria, archaea, viruses, and eukaryotic microbes. This helps us stay healthy, but it also does have a huge impact on our health because if those aren't healthy, it can actually cause us to be sick. And in healthy adults, we roughly have about 1,000 different types of gut microbiome and bacteria within our digestive system. Like I said, it is integral to our digestion and absorption of the nutrients we get from our food and diet. It's also integral to helping our immune system function properly. What it does is it allows the immune cells to understand what is good bacteria and what is bad bacteria. So the gut microbiome in our gut is actually good bacteria, and any foreign pathogen, it helps it understand that it's a bad bacteria. A gut microbiome can also play specific roles in the development of diseases and conditions, so much so that right now it's mainly linked to colorectal, liver, and even lung cancer. As you know, celiac disease, non-alcoholic fatty liver. It's also linked to allergies, asthma, and of course, obesity, cardiovascular disease, and neuropsychiatric diseases such as autism or ADHD. Most recently, studies have been looking into hormone-induced cancers, which include breast, ovarian, and endometrial cancers, to see if there is a link between that cell proliferation and gut microbiome mutations. And talking about mutations within our gut microbiome sequencing can happen between our eating habits and diets, environmental and or genetic factors that can alter those sequencings, specific medications that doctors prescribe based on health conditions, and even our bowel functions and movements can affect how our gut microbiome continue to develop over our lifetimes. So let's talk about the development of gut microbiome. So development starts to begin during delivery and continues after several years after we're born. So Enterobacteriaceae, Enterococcus, and Streptococcus are those first gut microbiome bacteria that develop to help jumpstart the rest of those species that will eventually develop over time. Delivery methods can actually delay development. Once the baby enters the vaginal canal, it actually jumpstarts the development of the gut microbiome because you're going through a sterile environment to a non-sterile environment. So that can actually jumpstart our gut microbiome. But if babies are born via C-section, there's actually a delay because that gut microbiome can't start developing, which actually leads to more infants being sick. You see more diarrhea, you see more less eating, just different types of health issues with these babies who are born via C-section. Diets, environmental factors, and genetic mutations can also disrupt the gut microbiome development. Like I said, different types of foods, different types of toxins within the environment, and even just genetic mutations that happen during embryosis and development of the embryo can disrupt how this gut microbiome is developed. One of the main factors that we're going to look at today is how breastfeeding versus formula feeding influence our gut microbiome development as we begin life. Both can either increase or decrease the production of specific gut microbiota. It just depends on what ones they are. So a few examples that we're going to see today is Lactobacillus, Pasteurellacea, Biophytobacteriaceae, and Biophytobacterium are just some of the main species of gut microbiome that can be increased or decreased in concentration. So before we get talking into the whole breast milk versus formula feeding debate that most cultures have, let's talk about the ingredients that are within both. So breast milk really depends on the diet of the mother. It does contain protein, fat, lactose, carbohydrates, and specific vitamins. And then just to go through those ingredients, protein can decrease depending on 4 to 6 weeks. This also depends on the diet of the mother. In fat for breast milk, it does contain high palmitic and oleic acids, which studies have shown is significantly lower at night. So when mothers breastfeed at night, babies are getting less of that fat. Lactose is always one of those that's in milk, so it's the principal sugar source, and it's there's more lactose found in the higher concentrations of those who produce higher quantities as they breastfeed. And of course, there's always those carbohydrates. You're going to have the oligosaccharides, depending on the stage of lactation and the genetic factors within the mother, how they produce breast milk. And then our typical vitamins B1, B2, B6, B12, D, and iodine are just some of the ones that are in there. For the formula brand that we're going to look at, it's called Similac. It's one of the most popular ones here in America. They do have protein, which helps power growth, fat and fatty acids, which includes DHA to help brain development, lutein, which helps the eye development, carbohydrates, energy, and fuel growth, HMO prebiotics, vitamins E, A, and D, and then minerals, iron, calcium, phosphorus, and zinc. And then this one has a special little mix that they include that has a blend of DHA, lutein, and vitamin E, which is particularly found in breast milk. But a lot of formulas try to mimic breast milk. One thing that we want to add as we're going to look at the rest of this analysis and presentation today is that most American baby formulas are classified as products with sugar added. Most of them are not on the labels of these formulas, and most of their grams per servings are measured between 1.28 grams and 11.16 grams, depending on the formula. So the big debate, breastfeeding versus formula feeding. Infants who are breastfed typically tend to have a more stable gut microbiome population. When they're supplemented with formula, their patterns start to change. They go from a breastfeeding pattern to a formula feeding pattern, which is also due to a wider microbiome development spectrum, which includes Clostridium, Streptococcus species, Lactobacillus, and Escherichia coli. So more of those gut microbiome develop in the presence of formula. And then once you introduce solid foods around 6 to 7 months, that also changes the breastfed infant's patterns to a formula-fed pattern. And then as we know, breastfeeding can have long-term health benefits, which means better immunity and better development of T cells. And it also lowers the risk of disease development, such as type 1 diabetes, irritable bowel disease, obesity, type 2 diabetes, and cardiovascular disease. One thing that we want to note is that there is a uptick in formula feeding due to a few factors within our culture here in America. One is being the lack of experience. Breastfeeding really isn't talked about a lot anymore, and a lot of women do not have the experience to do it. There's not enough opportunities to communicate with mothers who are breastfeeding. It's been one of those taboo things here in our culture, so it's something that they don't really get the chance to talk about. There's a lack of up-to-date instruction, and there's also sometimes shaming in mothers who don't understand how to breastfeed. And then hospital birthing practices are tied to making it difficult for mothers to actually exclusively breastfeed because the baby isn't latching right away. They often just jump to supplementing with formula. And then, of course, the lack of a combination for mothers who work to breastfeed or pump while at work. So our hypothesis for today's analysis is going to be, how does gene expression within our gut microbiome is dependent on specific factors such as diet. So how does breastfeeding versus formula feeding in infants affect this gene expression that actually ultimately keeps us healthy? So we're going to test this by looking at a DNA microarray, which is one way to analyze mRNA. While we're looking at this analysis, we're going to have to make sure that we keep asking these questions in our head. How does this gene expression lead to the development of diseases in the future? And how are specific ingredients, such as, like I said, the added sugar in formula, contributing to these mutations in gene expressions? So let's talk about DNA microarray. What is it? It's one of these analyses that allows us to analyze thousands of genes and their expression within specific DNA sequences. While it does say DNA microarray, it actually measures the different levels of RNA in gene products. And to test this, they actually use a rectangular platform with DNA spots, as you can see in the picture below. And that each little spot represents a different amount of DNA gene expression within those sequences and RNA. So that way they can get a good amount of information from each of those spots. There are some benefits to using microarray. It allows us to determine what genes have decreased or increased expression within each sequence that we're looking at, or such as this, we're going to be looking at gut microbiome. It allows us also to customize arrays for specific genes and features of interest. So you're allowed to see, like, okay, how does this gene express compared to this gene? And you're looking at specific genes that contribute maybe possibly to cancer or possibly to a different phenotype that is expressed within our bodies. There's fast results with this, so you do get your results rather quickly. And like I said, it is promising for cancer treatment, as it allows us to see how expression of these genes allows cells to proliferate into those cancer cells that we see. So there's different processes for microarray. One of them is the cDNA microarray, which is also known as the two-color microarray. It uses spotted cDNA that is often cloned. You amplify this DNA fragment and RNA fragments by using PCR, and then it helps, like I said, calculate the ratios of mRNA that bind to each spot. So if you look over to your right, you can see the little microarray panel with all the different colors. Each little color represents a different type of expression, the amount of RNA, and the binding factor that it has to these specific gene expressions. There's another one that's known as oligonucleotide arrays, or alpha-matrix arrays, that is known as a one-color microarray. It uses about 20 to 25 base oligonucleotides that are synthesized that allow us to see the expression within a certain type of gene or sequence. And then it's labeled and scanned in a single color. So that one is on the bottom right of this slide. As you can see, there's a little gene chip that it uses, and it's one single color, it's blue. And then the amplified picture shows the different expression rates in the blue with the red and the green, to show how the ratio of or mRNA. The difference between the two is alpha-matrix really doesn't read a competitive stuff, so it's not really reading all the different types of expression or hybridization between the two samples. Direct readout of RNA is in the form of a single sample within both the CDA microarray analysis. And here are some of the steps to the microarray. You want to start your study design and sample collection, and then you're going to get your patient sample. You're going to get the total RNA, and you're going to extract the cDNA from that. And then you're going to label it. You're going to amplify the signal by putting it through PR. Then you're going to get the fragment and label the fragments. You're going to put it on your gene chip. You're going to hybridize and watch those RNA sequences, and then you're going to help amplify the signal by using PCR. You'll scan it via computer. It'll image and process the image so you can see the analysis, and then you're going to put it into one of the many databases that are offered to show the sequencing for these steps. So today we're going to talk about the NCBI's Gene Expression Omnibus database, which allows us and provides the necessary tools to look at different experiences using different microarray analysis and sequence-based data products or procedures. Today we're going to look at dataset GSE 31075, which is the mutualism between gut microbiota and the host as revealed in a comparative study of breastfed versus formula-fed infants. So this analysis used 12 3-month-old infants. Their fecal samples were tested and used for intestinal mRNA, gene expression, and microbial DNA using microanalysis. Samples included 3 infants who were all breastfed and were all male, 3 infants who were supplemented with breast milk, which included 1 female and 2 males, and then 6 infants who were exclusively formula fed, which included 2 females and 4 males. The other, some of the other data that was in this experiment that I kind of want to go over to give you some more background before we look at the analysis is they analyzed the maternal age between both breastfed infants and formula-fed infants. And as you can see, the formula-fed infants were mothers were a little bit older. They analyzed their length at birth. Roughly, the breastfed infants were a little bit bigger at birth. Body weight, breastfed infants had a higher body weight. And then the intake up until 3 months of when this analysis was done, as that breast milk intake was about at 129 milliliters kg per day. Formula intake was at 134.8 milliliters of kilograms per day. So there's a little difference on the intake between both the breastfed and formula fed. The RNA-seq normalized count matrix that's used for this analysis was normalized by sequence depth and gene length. And then TPM, or transcripts per kilobase million, were used to analyze the expression value for this data set. So we're going to go ahead and get started by using the GEOR2R analysis, which allows us to look at the distribution value of the selected samples and all that stuff by using likelihood ratio tests due to there being more than 3 samples. So to start, we're going to visit the NCBI website. So the first thing you want to do is you want to look at the NCBI website. And over here, as you can see, you can either enter the GSE ascension number up here, or you can go down to gene expression and click there, and then click gene expression Omnibus to take you to go look at these different microarray analysis. So what we're going to do is we're going to go ahead and we're going to start here. So you're going to click here and you're going to click geo expression when you pull the pull down tab, or GEO datasets. And then you're going to enter the ascension number for the data analysis subset that we're going to look at is GSE 31075, and you're going to click search. So the first one that comes up is the case analysis that we're going to look at is mutualism between gut microbiota, and we're going to analyze this with the GO2R. So first thing you want to do is once you do that, it's going to bring us to this. You want to define the groups that you want to look at, because as you can see, there's 3 different groups that we have. So we want to make sure that those are all separated and defined. So you'll click on define groups. And then what you're going to do is you're going to type in the 3 different categories. So breastfed, breast milk supplemented, and formula fed. And then what you'll do is you'll click on each one and you'll highlight it and click on the category, and it'll automatically assign it to that category. So as you can see, breastfed is green, breast milk supplement is in purple, and formula fed is in pink. And once you have all of those analyzed and separated by looking at what ones are which, you're going to scroll down and you're going to go ahead and you're going to click analyze. And what that's going to do is that's going to analyze all our information. It's going to separate it in how we want it, and it's going to pull up a screen like this. So you're going to see a whole bunch of different types of graphs. You're going to see a volcano graph, a mean distribution graph, Venn diagram, box plot, all these different types of graphs, and then you're going to see data down here. So the first thing that we're going to do is we're going to talk about the different graphs that you see up here. There's 4 different graphs that I want to talk about. There is a box plot, which allows us to view the distribution values of our selected samples. So as you can see, we have breast milk supplement, and formula fed. Volcano plot, which displays the statistical significance of the negative log 10 p-value. You have a mean difference plot, which displays the log 2 folding change versus the average log 2 expression values. And then our Venn diagram that shows the overlap in genes that are significant to each other. And then also you have after you look at the graphs, you have these three columns that you want to look at. You have the adjusted p-value, you have the p-value, and you have the f. Because there were multiple samples, we are actually going to be looking at the adjusted p-value rate, because this one is, we want to look at this one as it's due to the multiple testing that we've done. Since there was 12 infants tested for this analysis, this is the adjusted p-value that we want to look at. Whereas you can see the p-values for each of these genes expressions are very low. Another thing that we want to do is we want to click this little thing that says select columns, because once we do that, we're going to be able to select these functions right here, the go function, the go process, and the go component. And then once you select that, you're going to hit set. And what it'll do is it'll add these three extra columns over here, so that it allows you to see what the processing is for each of the genes that are expressed or identified within this analysis. It tells you the function, it tells you the process, and then it tells you where these genes are expressed. So we're just going to go ahead and look at a few genes that we're going to see. So once you want to analyze a gene, you can click on the bar, as you see I've highlighted here, and it'll show you the sample value graph. So it shows the difference of expression between breastfed, breast milk supplement, and formula fed. As you can see, this one is mainly expressed in breastfed babies. And if you look up here at the highlighted stuff, you can see what the gene is. You can see the gene ID. Then you can see what it is. It's a tetratrisulfopeptide repeat domain 37. It is a protein binding gene, and it's located in the SCI complex. So then we have a few more genes. We have another one that shows breast milk supplement. We have another one that is formula fed. And then we have one that shows expressions between breastfed and formula fed. And you can click on all these genes and they'll have a little graph that shows each one of that. And then this one, I just want to point out that one thing that I did notice that we'll talk about later after we go and analyze the results, is that this one is a Tata box binding protein. And if we know from genetics, the Tata box is pretty important for mRNA translation and all that. So we will talk about that in a minute. So let's go ahead and analyze the results that we found by looking at this microarray analysis. It showed us about 250 genes that were mainly displayed within this analysis. If you want to look at all the genes, you can download the full spreadsheet, and it'll give you about the 1,000+ genes that they found within this experiment. In the ones that we have, 132 genes were identified, where 118 genes were unidentified. So those are those empty boxes that you see right here. I'll just go back. So if you see you have IRF1 and then TAF15, you have those empty boxes right there. Those are unidentified genes. And then throughout looking at all the different graphs of the 250 genes, we noticed that male infants had a higher gene expression in breastfed and breast milk supplements. So overall, breastfed babies had more, males had more gene expression, whereas those who were exclusively formula fed showed higher expression in female infants. And then overall, formula fed infants actually showed lower gene expression relative to the breastfeeding samples, which was actually kind of surprising due to all the extra added ingredients within formula. And then here's just a distribution of out of the 250 genes that I did. I counted all the distributions expressed. So for breast formula, or for breastfed, you have 20 total genes that were highly expressed. BMS, the breast milk supplement, there was over 100. So in total, you had 120 genes that were expressed between breast milk. For formula fed, there was only 56. For breastfed and breast milk supplemented together, where they were expressed at the same time, there was about 52. For breastfed and formula fed, there was about 19. For breast milk supplement and formula fed, there was only about 3, which I found that was really, really surprising compared to breastfed and formula fed, which was 19. So there's about a 16 gene expression difference. And then there was none when all 3 were expressed. So the one thing that we want to analyze is that after multiple testing, I talked about the adjusted p-value for each gene that ranged from 0.128 to 0.159, which is the significance of that. And you just want to make sure that it measures the probability of obtaining the observed data. And then we have our F statistic, which actually combines all the T statistics that you would see normally in a microarray analysis. And this represents the overall test of significance which is we want to make sure that our observed genes are matching our hypothesis. And those ranged anywhere from 9.6 to 38.4 within gene expression. So some of the genes that I just kind of wanted to go over as far as expression within everything is, you can see that we have TTC37, which is the tetratriisopeptide, repeat domain 31. It is a protein binding gene, and as you can see, its adjusted p-value was 0.59 after multiple testings, and its test of significance was only a 10.92, which was one of the lower ones compared to some of the other ones that we saw. We do have LOC 643733, which is a capsase 4 apoptosis-related cysteine peptide pseudogene. You can see that it's more common in breast milk supplemented with a small adjusted p-value of 1.28, and its frequency was 32.42. As you can see, these three, their frequencies were very high. And so were their adjusted p-values, which means that they were more expressed within these ones as they are more important. You have mucin 15, which is a cell surface associated oglycine processing. And then, like I said, you have the Tata box one, which is the binding protein, which allows for, DNA, nucleotide, RNA, and protein binding, which is very important for mRNA translation. So how does this research results impact future research? So we could actually allow additional microarray analysis to be performed, and this can actually help us identify those unidentified genes that we saw within the analysis. As we did see, there was 118 unidentified that we do not know what their function is, we do not know what they are, but they were expressed within breastfeeding, breast milk supplemented, or formula fed. It allows us to, by performing additional analysis, we can measure gene expression based on the formula brand. In this analysis, it doesn't specifically mention a formula brand, but because there are so many formula brands out there with different amount of ingredients, different amount of sugar added, we can see how each formula brand can impact gene expression within the infant's gut microbiome. It also displays how both our bodies and gut microbiome react to different dietary factors. As you can see within the expression, most of those genes were expressed with breastfeeding, but there were some that were still expressed with formula feeding, and some of those can actually help decrease the chance of cancer, while other those can increase the risk of developing diseases and so as cancers. And this just allows us to see how, like I said, remember to think about that question of added sugar can affect the development of infant and gut development of the infant's gut microbiomes, which include nucleotide changes that create mutations within these sequences that allow these gut microbiome to develop differently. And then also, how does it affect our gene expression? By looking at the different sequences while exposing them to these sugars, such as, corn syrup, added glucose, maltose, fructose, anything that's added into these baby formulas, how does that change the expression? What do we need to look for within these sugars and our gut microbiome for infants? And then further analysis can also help identify genes within the gut microbiome, like I said, that can lead to disease and cancer development. By identifying these mutations and possible causes, it can allow us to help identify cancers that are known as silent cancers. Most cancers, you do have symptoms, but there's some like colorectal cancer and ovarian cancer where if you don't catch it in the early stages, you won't know about it until it's in stage 3 or 4. And currently there's no testing for early stages of those cancers. So understanding how our gut microbiome develops and how even being fed in the early stages of our life can affect how these cells proliferate into cancer cells and hopefully help with early treatments, early diagnoses for specific diseases and cancer. So to summarize, gut microbiome play a crucial role in our health and can determine how diseases develop, such as cancers, colorectal, and even cardiovascular diseases, obesity, type 2 diabetes. There will always be that debate about breastfeeding versus formula feeding because it also depends on culture, society, and how people view feeding their infant. You do have those mothers who are sometimes all about breastfeeding. So there is that controversy, but it'll always have that debate. We want to make sure that we get all that information out there. Microarray analysis allows us to see the importance of gene expression and how this can lead to our health or development of disease. And while there's a rise in formula feeding, gene expression was still more prevalent in breastfed and breast milk supplemented infants, which is actually surprising given all the added ingredients in formula. And then by identifying genes expression early, we can use this data to help eventually pinpoint gene mutations that lead to specific diseases and cancer development. Other than that, I just wanted to show you all my references that I used for this presentation, and then I wanted to thank you for watching my presentation. If you have any questions, feel free to post them in the discussion board underneath this video, and I will be happy to answer them. Thank you once again for watching my presentation.